This is part 6 of the chapter 2 lectures. Hopefully we're nearing the end. We left off with, we still had a little bit left to go with our macromolecules, the category of lipids. We have not covered phospholipids, and that's important. So phospholipids are lipids that are found that form the backbone of the plasma membrane in our cells, or sometimes called the cell membrane. And phospholipids are composed of a glycerol, like we saw in the triglycerides. And instead of 3 fatty acid tails, there are 2 fatty acid tails in phospholipids. And in addition to that, there's a phosphate head, or there's a phosphate group that's attached as well. And we call that the head of the phospholipid, and the tails are the fatty acid tails. So the phosphate head of the phospholipid is polar and soluble in water, and the fatty acid tails are nonpolar and they are lipid soluble. So what happens is the plasma membrane is composed of a bilayer of these phospholipids, where the heads face outward and the tails face inward toward each other. We will cover this a little bit more in chapter 3. But phospholipids, for now, just know that they're found in the plasma membrane of cells. Okay, let's see. Let's look at number 21, describe lipids. Remember, lipids are similar to carbohydrates in that they're composed of carbon, hydrogen, and oxygen, but the ratio of oxygen to carbon and hydrogen is much lower. Lipids are typically nonpolar and insoluble in water. 22, which kind of lipid would be found in a sample of fatty tissue taken from beneath the skin? Remember, we learned that body fat is called triglycerides. And then 23, which lipids would you find in human cell membranes? And that would be phospholipids. Proteins are the next category of macromolecules. They're the most abundant organic molecules in the human body, and they account for about 20% of the total body weight. Now, they're still going to be composed of the same 3 elements as lipids and carbs: carbon, hydrogen, oxygen. But then there's also going to be nitrogen, and sulfur and phosphorus may also be present. And you may recognize all of these elements as the same 6 elements that I had you learn from the, the elements on the periodic table, that you have to know these names and symbols. They're all found in proteins. As far as functions of these proteins, they have many, many functions, at least 7. Proteins can function in support. For example, the protein collagen is a protein that's known for its strength, and so it's found in bones and skin and other areas. Proteins can function in movement. The proteins that cause muscle contraction, for example. Proteins can function in transport. One example there is hemoglobin, which is found on red blood cells, and it transports oxygen throughout the blood. Proteins can function in buffering. And remember, a buffer is any chemical that helps to remove hydrogen ions or add hydrogen ions to solution in body fluids in order to maintain a constant pH. Proteins are used in metabolic regulation, regulating metabolism. The chemical reactions that occur in the body can be done by enzymes. And coordination and control. Some of our hormones and neurotransmitters in the nervous system are proteins. They're protein-based. Now, we do have hormones that are lipids, like steroid hormones, but we also have hormones such as insulin and growth hormone that are proteins. And then proteins can function in defense. I'm sure you've heard of antibodies. These are proteins produced by our immune system. So those are just some examples of protein function. And I just went through all of this, so I should have done it with the notes, but anyway, I'm not going to repeat it. I just want to make sure the examples I gave you are the same as they are here. So metabolic regulation, the examples they gave you were enzymes that speed up the chemical reactions in living cells, hormones, and I think I added one, neurotransmitters can also be proteins. And then in defense, we have antibodies, and then an additional one for defense are clotting proteins that help to produce blood clots when we're bleeding, when we have an injury. All right, you need to know this structure right here. You need to be able to recognize that this is the structure of an amino acid. An amino acid has a central carbon, and you may have noticed by now that carbon can form 4 covalent bonds. So one of the covalent bonds is to a hydrogen atom. The covalent bond on the right side of the page here is a carboxyl group. So this is the acid part of the amino acid. The covalent bond on the left is the amino group, which is a nitrogen atom bonded to 2 hydrogen atoms. This is the amino part of the amino acid. And then you see a covalent bond to an R group. And R is not the symbol for an element on the periodic table. It actually means a variable chemical group. So it can be a single element, or it can be a group of chemicals. But for each different amino acid, and there are 20 different amino acids in the body, each R group is what makes the amino acid different. So make sure that you recognize this structure as the structure of an amino acid. And then proteins are chains of amino acids, and they form by amino acids bonding to each other through what's called a peptide bond. And that's when the amino group of one amino acid bonds to the carboxyl group of the other. So peptide bonds are another bond that we're learning now that link molecules together, and this time they form proteins. Okay, so peptides are amino acids, chain of amino acids connected by peptide bonds. If we only have 2 amino acids in the chain, it's a dipeptide. If there's 3 or more, we call it a polypeptide. 100 amino acids or more, that's when we call it a protein. So the difference in a polypeptide and a protein is just the number of amino acids in the chain. You do need to know the 4 levels of protein structure. You'll need to be able to name them. They are primary, this is very easy, primary structure, secondary, tertiary, and quaternary. The primary structure of a protein is literally just the sequence of amino acids. So the chain of amino acids is the primary structure. Then that chain or sequence of amino acids can form either into an alpha helix, which is like a spiral shape, kind of like DNA, or a beta sheet, which is pleated. If you've ever taken a piece of notebook paper and folded it into a fan. So a beta sheet is pleated in structure, and an alpha helix is spiral in structure. So a secondary structure is either the alpha helix shape or the beta sheet shape. And here's an image so you can see the primary structure is represented by 9 different amino acids here. The secondary structure is when that chain of amino acids either spirals into an alpha helix or folds into a pleated beta sheet. And then the tertiary structure is when the secondary structure, the alpha helix, actually forms further into a 3-dimensional structure, a more complex folded and twisted shape specific to that protein. That's the tertiary structure. And this is the image I want to give you so that you can kind of relate. So if you've ever seen a phone cord, I hope you have. I know a lot of our phones don't have cords, but there are still some that do. So a phone cord is like the cord itself is shaped like an alpha helix. But if you took that cord and you tied it into knots and you folded it and you formed it into a really complicated shape, that would be similar, analogous to the tertiary structure of a protein. And then let's say that you did that with 4 different phone cords and then you hooked them all together. That would be analogous to the quaternary structure. It's when you take several 3-dimensional structures, several tertiary structures, and link them together. So hemoglobin is an example of a protein that has a quaternary shape. And this is just the definition. The tertiary structure is the complex coiling and folding, giving the 3-dimensional shape of the secondary structure. And then the quaternary structure is an interaction between polypeptide subunits. So in other words, interaction between various tertiary structures, and they form an even more complex protein. Proteins can be classified as fibrous proteins, as globular proteins. Hemoglobin is a globular protein. Fibrous proteins typically help maintain structure in the body. Denaturation, you need to understand the information on this slide because there is, you definitely need to be able to explain why it is important for a protein to have a specific shape and specific tertiary and quaternary levels of structure. Is because if this shape changes, we call that denaturation. It's the change in the 3-dimensional shape of the protein. So change in tertiary structure, which would also change the quaternary structure. But when that structure or shape changes, it can result in a non-functional protein. So this is another example of where structure determines function. If the structure is not exactly as it should be, as it was designed to be, then the protein can't do its job. And this can lead to death. I mean, it can cause, like it says, it can cause irreparable damage to organs and organ systems. So another thing is the reason for denaturation can be temperature. A lot of times it's because the temperature is too high, but it can be low temperature as well. pH, when the pH is too low or too high, and the ions that are present in the body fluids. So these things can change the composition of, can change the shape of the protein. And we call that denaturation, and it will make the protein non-functional, okay? Enzyme function, remember that enzymes are protein catalysts, so they help to speed up the chemical reactions that occur in the body. The chemicals that the enzymes react with are called substrates. So they're the reactants in the chemical equation. And what they do is the substrates bind to a place on the enzyme called the active site. And when they bind like a lock and a key, that changes the shape of the enzyme temporarily, and then that causes the formation of the product. That same enzyme can continue to catalyze another reaction, but the enzyme is specific. There's specific enzymes for specific substrates. So here's an image that shows you how it works. The substrates are chemicals that are just labeled S1 and S2. They bind to the active sites on the enzyme. Notice that there's specific shapes. And then that enzyme changes shape and forms the product, which is released. And that same enzyme can go back and react with S1 and S2, with more S1 and S2 chemicals. How does boiling a protein affect its structural and functional properties? It denatures the protein. It changes its structure, its tertiary and quaternary structure, the 3-dimensional shape of the protein, and then that causes it to be non-functional.